Hello, everybody. I hope you're having a good day. Before we start tackling our topic today, let us introduce our group first. We are the House of Joseph from 11 Esther that will discuss to you guys the topic about electron transport chain. So let's now dive deeper with our topic today.
What is electron transport chain or ETC? The electron transport chain or ETC in eukaryotic cell is embedded in the inner mitochondria membrane, while in prokaryotic cell it is located in the plasma membrane. This chain is composed of many protein complexes and electron carriers designed primarily to facilitate electron transfer from molecules like NADH and FADH produced during earlier respiration stage to oxygen, the final electron acceptor. These electron transfers generate a proton gradient across the membrane, driving ATP production. The electron transport chain is the final stage of cellular respiration and is highly energized efficient, producing most of the ATP in aerobic organism and ADH and FADH form in earlier processes such as glycolysis and Krebs cycle to oxygen, the final electron acceptor. What are the importance of ETC producing ATP? For aerobic organism, the ETC is essential for producing ATP. As electrons move through the chain, energy is released which pumps proton across the membrane, creating a proton motive force. This force drives ATP synthase to convert ADP and inorganic phosphate into ATP. Unlike glycolysis or the Krebs cycle alone, the ETC is dramatically boosts ATP yield, accounting for about 34 of the 38 ATP molecules generated from the one glucose molecule during respiration. By releasing energy in measured steps, the ETC avoid a large harmful energy release that could damage the cell. As electrons move along the ETC, energy is released in stage. This energy is used to pump proton across the membrane, forming an electrochemical gradient known as the proton motive force. This gradient powers ATP synthase, an enzyme that uses the flow of protons back across the membrane to synthesize ATP from ADP and inorganic phosphate. By controlling and by controlling the release of energy through steps, the ETC yields a high amount of ATP, around 34 of the 38 ATP molecules generated from a single glucose molecule and aerobic respiration while avoiding a sudden energy release that could damage the cell. This efficient ATP producing provides aerobic organism which is a significant energy advantage over anaerobic pathway.
But what is NADH and FADH2? The NADH and FADH2, they are the carriers of the electrons. These are the byproducts of the earlier stages of cellular respiration such as the glycolysis and the Krebs cycle. And also, take note that Krebs cycle is also known as citric acid cycle. Next, these two molecules form the key components of ETC or the electron transport chain, being the last process of the cellular respiration and taking place inside the inner membrane of mitochondria. And now let us tackle the two electron carriers molecules and let us now start with the NADH. It is also known as nicotinamide adenine dinucleotide, and this electron carrier is an energy-rich electron carrier formed mainly during the glycolysis and the Krebs cycle. NADH carries electrons and the hydrogen ions to the ETC, or what we call the electron transport chain, where it finally donates its electrons. When NADH transfers its electrons to the ETC, it becomes oxidized to become NAD+, again, and in this state, it is reused in earlier stages of cellular respiration. And take note that every molecule of NADH supplies a pair of electrons to the ETC and thereby greatly contributing towards the energy produced within the cell. So basically, the job of NADH is to provide or supply a pair of electrons to the process of ETC, or what we call the electron transport chain. After having the NADH, of course we also have the FADH2, also known as the flavine adenine dinucleotide, and it is just another electron carrier just like the NADH. But it has a slightly lower energy level compared to NADH, and most of the FADH2 that gets produced occurs in the Krebs cycle and contributes electrons to the ETC later than NADH. And when FADH2 donates electrons, it gets oxidized to FAD and electrons are introduced into the ETC to assist in the production of ATP. And for this part, we could actually see the process of the ETC or the electron transport chain itself. And for the last part of NADH and FADH2, they are both the initiators for the ETC or the electron transport chain, functioning as the electron flow initiators. They pass these electrons to the ETC, which starts the domino-like cascade of electron transfer through several protein complexes that will be discussed later. Lastly, electrons movement generates chemical gradient across the mitochondrial membrane, producing ATP or what we call the adenosine triphosphate.
Now, let's dive into the structure and key components of electron transport chain. For the first one, we have complex 1, also known as NADH dehydrogenase. It absorbs electrons from NADH, which is produced by earlier metabolic processes such as the Krebs cycle. NADH transfers electrons and is oxidized to NAD+. This complex uses energy from the redox reaction to pump 4 protons from the mitochondrial matrix into the intermembrane space, which contributes to the proton gradient required for ATP production. Electrons are transported to the ubiquinone and reduced to ubiquinol. In complex 1, electrons are transferred from NADH to ubiquinone through flavin mononucleotide and iron sulfur clusters, leading to the reduction of ubiquinone to ubiquinol and the pumping of protons into the intermembrane space. Secondly, we have complex 2, also known as the succinate dehydrogenase. Complex 2 gets electrons from FADH2, which is created during the Krebs cycle when succinate is converted into fumarate. Unlike complex 1, complex 2 does not pump protons across the membrane, hence its contribution to the proton gradient is less important. FADH2 electrons are transported to ubiquinone, reducing it to ubiquinol analogous to complex 1. Complex 2 transfers electrons from FADH2 to ubiquinone through a series of iron sulfur clusters without proton pumping, producing fumarate and reducing to ubiquinone to carry electrons further along the chain. Thirdly, we have complex 3, commonly known as the cytochrome C reductase. It takes electrons from ubiquinol and transfers them to the soluble electron carrier cytochrome C. This complex includes a process known as the Q cycle, which efficiently pumps protons across the inner mitochondrial membrane, contributing to the proton gradient. Specifically, complex 3 transports 4 protons into the intermembrane gap for every 2 electrons that pass through. In complex 3, electrons are transferred from ubiquinol to cytochrome C, releasing protons into the intermembrane space, while cytochrome C transfers electrons to complex 4, facilitating continued electron transport and proton gradient formation. Last but not the least, we have complex 4, also known as cytochrome C oxidase. Complex 4 is the last complex in the electron transport chain. It accepts electrons from cytochrome C and transmits them to the terminal electron acceptor molecular oxygen, reducing it to water. This mechanism is linked to the pumping of protons into the intermembrane gap, which increases the proton gradient. Complex 4 translocates 2 protons for every 2 electrons transported, completing the chain of events that creates the gradient required for ATP synthesis. In complex 4, electrons are transferred from cytochrome C to oxygen, the final electron acceptor, through a series of redox reactions involving cytochromes and copper centers. This process drives the pumping of additional protons into the intermembrane space and results in the formation of water, further contributing to the proton gradient used for ATP synthesis. Next we have electron carriers. The first electron carrier is the ubiquinone, or coenzyme Q. This lipid-soluble molecule transports electrons between complex 1 and complex 3 as well as from complex 2 to complex 3. Ubiquinone can exist in both oxidized and reduced forms, allowing for efficient electron transfer across the membrane. Also, we have cytochrome C. Cytochrome C is a tiny soluble protein that transports electrons from complex 3 to complex 4. It can only take and contribute one electron at a time, making it an important intermediary in electron transport process.
Proton gradient in ATP synthesis. Proton gradient is a difference in proton concentration across a membrane. It plays a key role in cellular energy productivity. This gradient was formed when protons are distributed in the membrane called electrochemical gradient. This occurs with the electron transport chain that is transferred to protein complexes. ATP synthase is a membrane-bound enzyme complex that have the energy from the proton gradient to give ATP. It uses chemoosmosis to synthesize ATP using proton gradient. Proton gradient refers to the gradual dispersal of proteins due to the frictional resistance, increase of the geometries. In the electron transport chain, a cluster of protein transfers electrons through a membrane within mitochondria to form a gradient of protons that drives the creation of adenosine triphosphate. In fasciolated diffusion, molecules diffuse across the plasma membrane with assistance from the membrane proteins such as channels and carriers.
ATP synthase is an enzyme that catalyzes the formation of the energy storage molecule adenosine triphosphate using adenosine diphosphate and inorganic phosphate. ATP synthase is a molecular machine.
Without oxygen serving as the final electron acceptor, the ETC cannot operate as intended. Because oxygen keeps the process clear by removing spent electrons, so new electrons can continuously enter the chain. Because the proton pumping necessary for ATP synthesis is driven by each stage of electron transfer process, the production of ATP depends on this constant flow. The ETC would buck up in the absence of oxygen, stopping the ATP synthesis and cutting off the cell's primary energy source, which would ultimately affect the cellular survival and function.
The electron donors are NADH and FADH2. They donate electrons, thus starting the process. NADH and FADH2 produce an earlier stage of cellular respiration, donate electrons the ETC, and initiating the process. Electron transport and proton gradient. In ETC, a series of protein complexes move the electrons, thus producing a proton gradient across the mitochondrial membrane. As the electrons move towards the ETC, they are passed from the one protein complex to another. This movement of electrons release energy, which is used to pump protons H+ from the mitochondrial matrix to the intermembrane space, creating a proton gradient across the inner mitochondrial membrane. ATP production. The proton gradient drives the ATP synthase reactor that produces ATP. The proton gradient represents a form of stored energy. This gradient drives ATP synthase. An enzyme thus uses the energy from the proton flow to synthesize ATP from ADP in inorganic phosphate. Role of oxygen. serve as the last electron acceptor reacting with protons and electrons to form water, an essential component for ETC function. Oxygen acts as the final electron acceptor in the ETC. It combines with electrons and protons to form water, a byproduct of cellular respiration. This process is essential for the continuous flow of electrons through the ETC. Vital for cellular respiration, ETC is known to be the final phase of cellular respiration and produce most of the ATP used by cells. The ETC is the final stage of cellular respiration, the process by which cells break down glucose to generate energy. It produces the majority of ATP, the cell primary energy source, making essential for cellular survival and function. Energy for life. Basic cellular activities ranging from muscles contraction to repair and growth cells. ATP produced by the ETC powers a wide range of cellular activities. Contract is given to basic activity. Next is cell repair and growth is essential for maintaining and building tissues. And last is base cellular processes, includes protein synthesis, DNA replication, and active transport. Implication for health. Energy associated with mitochondrial diseases and several aging-related conditions result from malfunctioning in the ETC. Malfunctions in the ETC can lead to energy deficiencies, causing various health problems. Mitochondrial diseases, a group of disorders affecting the mitochondria, often resulting in muscle weakness, fatigue, and the symptoms. Aging-related condition. As we age, mitochondrial function declines, contributing to age-related diseases and overall decline in health. Scientific and medical application. The knowledge about ETC leads to therapies and metabolic disorders and bioengineering applications such as developing efficient biobatteries. Understanding the ETC has opened up new avenues for scientific and medical advancement. Therapies for targeting the ETC can help treat conditions like diabetes and obesity. Bioengineering application. The principles of ETC are being used to develop efficient biobatteries which could revolutionize energy storage and production.
That concludes our discussion today. Thank you for listening to the House of Joseph. We are all hoping that you've learned something new today. See you around!